It is one of the important step in the process of biotechnology or DNA fingerprinting. Whenever the DNA content is lesser with us and we need to amplify, we use this technique. So it is done in various steps. The first step is known as denaturation, and which is done at 92 degrees Celsius. So in this process, the two strands of the DNA, because of high temperature, they move apart from each other because the hydrogen bonds are being broken. Now the next process is known as annealing, and annealing means to join. This is done as 52 degrees Celsius, and in this primers are being joined. You know that DNA polymerase cannot do the process of initiation, but it can do the elongation. So in annealing, short RNA primers are being attached. We know that DNA is antiparallel, so here it is going to be 3 prime, then it is going to be 5 prime. So always remember the end that is going to be the 5 prime end, their primers will be attached. So with blue color I have marked the primers. In the fourth and the last step, extension is going to happen. Extension means the elongation, right? This is being done at 72 degrees Celsius, and very important part that just prior to the extension we require Thermus aquaticus polymerase. This is the bacteria which has the capacity to live at extremely high conditions, and the short form is taq polymerase. From thermus we have taken T, from aquaticus we have taken AQ. Deoxynucleotide triphosphates are being taken, magnesium ions are also being taken, because of which the long stretch is formed, right? So here you need to write these red strands that are being formed are the newly extended chain of DNA, which requires DNA polymerase, which is being extracted from Thermus aquaticus, because at high temperature of 72 degrees Celsius normal DNA polymerase does not act. Along with this deoxynucleotide triphosphate, so suppose here it is AT, so bases are to be given, right? That's what it is written, deoxynucleotide phosphates. So opposite of A will be T, opposite of T will be A. So this is how nucleotides will get arranged and long stretches of DNA is being formed. So polymerase chain reaction is a very important step of DNA fingerprinting. Whenever DNA content is less and we want to increase, it is being used. And in biotechnological process also, if the gene of interest is lesser in amount and we need to increase its content, then PCR is done.